Today, we're going to discuss the basic concept of the technology, background of development, carcinostatic potency, benefit of use, and summary of all the above. As we all know, the technology is all about microRNA suppression of mRNA, with the target gene related to growth of the specific tumor called FOSL1. As you can see, the seed sequence of the specific sequence of 3' UTR of FOSL1 is recognized by the microRNA called miR4516, and that's how the technology works complementarily. Upon such recognition, mRNA degradation takes place if under total complementarity, whereas protein translation occurs if under partial complementarity. You might be more interested in how we discovered miR4516 than the technology we developed. We also believe that understanding the development process makes our composition even more reliable when taking into account that cancer interacts with tumor microenvironment. We need to take a first look at fibroblast, where fibrosis takes place more often than not in all solid cancer types. Only frequency varies. When fibroblast does not interact with cancer, meaning that cancer is yet to train fibroblast, it is reported that cancer-related progression, such as tumor growth, is effectively inhibited. When cancer continuously interacts with fibroblast, meaning that cancer trains fibroblast so that the fibroblast eventually becomes CAF, that stands for cancer-associated fibroblast. It can be said that cancer ends up metastasized, promoting progression of cancer and giving rise to cancer progression to take place. According to study results, CAF is originated from the normal fibroblast existing near the tissues, and, as you all know, CAF eventually contributes to tumor progression. It is also known that the such CAF role is made possible by transitioning a specific type of micromolecule off the medium called exosome to cancer cells, and that normal fibroblast inhibits tumor progression on the contrary. This transcriptome of RNA, more specifically the expression patterns of the transcriptome, differ between CAF and normal fibroblast, as a number of authors say, implying that taking into account all of the background knowledge related to CAF, tumor progression might have affected the amount or composition of molecule while being transitioned within the exosome from normal fibroblast to CAF. Back to where we was, we're talking about microRNA that impacts transcriptome and thereby affects cell behaviors. A microRNA is capable of suppressing multiple target genes. Like all other CAF studies discuss, it is obvious that exosomal microRNA engages in tumor progression. It is worth mentioning, however, that some authors focus on drug tolerance. So, all in all, exosomal microRNA is capable of serving as a viable predictive marker and the therapeutic target at the same time. In our attempt to find out miR4516, we looked deep into the breast cancer tissue, more specifically, the specific breast cancer tissue as wide as 10 square centimeters that has no invasive trace. We've got the CAF extracted and successfully detached normal fibroblast off the normal tissue at about 10 millimeters away from the neighboring cancer tissues. Take a look at Figure B, where the exosome in cultured cells is secreted from cells to media. We verified the media to make sure CD63 is a viable exosome marker by checking the normal media and cultured media and confirmed that CD63 is overexpressed. The same viability was verified for ALIX and TSG101. Next, in terms of the amount of exosome off CAF and normal fibroblast, we've also verified that there exists no significant difference in the amount of exosome derived. Our study regards RNA, the microRNA. so we've verified the extracted microRNA is pure. Taking into account that microRNA suppresses mRNA, we focused on assessing the exact decreasing amount of exosomal microRNA in calf compared to normal fibroblast by way of most extraction microarray. See the Table A. We've checked 3 different microRNAs. And, in terms of fold changes, we verified that the decreasing amount of miR-4516 was larger than any other types of microRNA. We've also verified that, taking into account that isolation of fibroblast took place previously, such decrease in miR-4516 takes place more intensively in tumor tissues than normal tissues. As like I told you before, fibroblast transitions into calf based on the cancer cell training the normal fibroblast. We've worked the cancer's condition rhythm in an attempt to activate normal fibroblast. Compared for this were MDA-MB231, one of the most common invasive breast cancer cell line, and MCF7, one of the most common non-invasive breast cancer cell line. Decrease was seen in both cases, but only with statistical significance in MDA-MB231, meaning that the invasive breast cancer cell lines train fibroblast. Then, we needed to see if miR-4516 transitions via exosome in both normal fibroblast and calf. Doing so, we've worked with the conditioned media to treat cancer cells, followed by separation of exosome. See the figure D to see the significant amount of miR-4516 was observed in cancer cells in normal fibroblast, and the same result was derived when exosome is separated. As part of the supervised analysis to understand how microRNA interacts with cancer cells, based on the known mechanism of microRNA suppressing mRNA, first of all, these are the phenotypes related to cancer progression. We've sorted out genes that induce glycolysis, EMT, apoptosis, migration, and proliferation and studied how these genes are bound to microRNA. As a result, we've discovered that proliferation was induced in some of the major phenotypes. About 60 genes out of a total of 558 genes were related to tumor growth, accounting for the most. With that being said, we've studied whether microRNA decrease tumor growth rate in cancer cells. As a result, the growth of cancer cells in MCF7 and MDA-MB231 decreased with statistical significance. As you can see on the figure G that illustrates the expression in cancer cells, taking into account that cancer cells coexist with calf in tissues in clinical settings, we've drawn comparison with the condition where miR-4516 was expressed in calf, which simulates normal fibroblast. With calf, growth of cancer cells in MCF7 and MDA-MB231 decreased with miR-4516 expression compared to the control group, as demonstrated in the table below. This means that miR-4516 effectively targets growth-related genes. To get deeper understanding of the exact mode of action, we've worked on verifying the identity of genes. A total of 558 genes we previously discussed are categorized into either oncogenes or tumor suppression genes, where oncogenes are more likely to be associated with proliferation, meaning that they are more viable candidates. According to the categorization, in the intersection of oncogenes and proliferation genes, there were 16 genes. In the intersection of tumor suppression genes and proliferation genes, there were 7 genes. This means that more oncogenes are capable of adjusting tumor proliferation genes. To see how genes are correlated to each other, we've analyzed correlation of genetic expression. I mean the expression of those target genes after overexpressing miR-4516 in those breast cancer cells. Decreasing pattern was seen in these two cell lines, highlighted blue. It was confirmed that miR-4516 suppresses breast cancer cells in 3 different gene types: FOSL1, PAX5, and TCL1A. To reverse confirm the correlation between expression of miR-4516 and these 3 gene types, we've made sure that the reverse correlation exists, as seen in the figure, where miR-4516 decreases in the tumor zone while target genes increases. With both correlation and reverse correlation verified, we now need to confirm whether microRNA effectively suppresses and eventually binds the target genes by way of luciferase assay. As a result, the expressed miR-4516 effectively binds to the 3' UTR of the target gene. With the suppressive and binding capability confirmed, we now need to confirm again that expression of these target genes decreases in miR-4516 expressed in the exact cancer cells we've used in the above tests and assays. As seen in the figure, all 3 target genes decrease by the effect of miR-4516 in MCF7 and MDA-MB231 both in terms of mRNA level. In terms of protein level, to assess mRNA viability, we weren't able to get any viable result on FOSL1 with low expression in MCF7, quite similar result for PAX5 and TCL1A, as no FOSL1 result was acquired. On the contrary, we've seen drastic increase in FOSL1 expression in MDA-MB231, later controlled significantly by the effect of miR-4516. Just like that, we verify that miR-4516 effectively targets FOSL1 in, among other breast cancer cell lines, MDA-MB231. To verify correlation between FOSL1 and proliferation of tumor cells, we've treated FOSL1 with shRNA. The result implied decrease in growth of MDA-MB231 compared to the control group when expression of FOSL1 decreases. Taking into account that MDA-MB231 is one of the most common triple-negative breast cancer cell lines, we've attempted to check the potential cancer cell inhibiting effect in other triple-negative breast cancer cell lines. The significant increase in FOSL1 in BT20 and HS57AT was effectively controlled by the effect of overexpressed miR-4516. Decreasing both protein level and mRNA level, as well as tumor growth, with statistical significance. So far, we've talked about the background. Now we're going to talk about the pharmaceutical potency of miR-4516 by testing controllability of cancer cell proliferation by the effect of exosomal transfer induced by miR-4516, as well as a parity of effect between overexpressed miR-4516 and mimic treatment. We've also tested whether or not the reverse correlation between miR-4516 and FOSL1 takes place in other triple-negative breast cancer cells, whether or not expression of FOSL1 accompanies in other genetic alterations, whether or not the reverse correlation between miR-4516 and FOSL1 takes place in any other cancer cells, and whether or not FOSL1 can be overexpressed in any other cancer cells. To test the pharmaceutical potency of miR-4516 controlling proliferation by exosomal transfer, we started out by overexpressing miR-4516 in CAF. GW4869 inhibits, or in other words, blocks exosomal secretion. In CAF cells with overexpressed miR-4516, exosome is blocked. Upon treatment of GW4869 in the overexpressed miR-4516, the miR-4516 expression level in the cells did not change. While GW4869 treatment decreasing the amount of exosome secreted, we've also discovered that, despite some variations in statistics, the expression rate in calf cells with overexpressed miR-4516 was significantly higher than that in the normal fibroblast. As a result, the hypothesis of increasing growth of tumors when exosome is blocked upon co-culturing of breast cancer cells and calf cells with overexpressed miR-4516 with and without exosome secreted is very true, meaning that exosomal transfer effectively inhibits proliferation. We then moved on to check whether the miR-4516 mimic effectively decreases growth of triple-negative breast cancer cells it treats. As a result, we confirmed that the miR-4516 mimic effectively decreases growth of triple-negative breast cancer cells, all with statistical significance. We further moved on to check whether the miR-4516 mimic treats cancer cells in the actual tissue-like environment, meaning the calf cells instead of co-cultured environment. As a result, we confirmed that the miR-4516 mimic effectively decreases proliferation of cancer cells in the presence of calf in 2 out of triple-negative breast cancer cells. This means that the correlation between FOSL1 and expression of miR-4516 in triple-negative breast cancer cells is finally discovered, where in tissues with high FOSL1 expression, miR-4516 is expressed less, and vice versa. This reverse correlation also applies to miR-4516, where it is less expressed in triple-negative breast cancer tissues than in non-triple-negative breast cancer tissues. Check out on the right-hand side that FOSL1, which is the target gene, is less expressed in non-triple-negative breast cancer tissues than in triple-negative breast cancer tissues. A totally different story will begin where there exists any genetic alteration. When we further move on to understand which genetic alteration occurred in the target gene by analyzing TCGA database, we've confirmed that mRNA of FOSL1 was overexpressed with statistical significance in triple-negative breast cancer tissues, where major genetic alteration of FOSL1 is amplification, as you can see, which is why we still think that overexpression is the major issue that is worth attention. As for FOSL1, patients from the groups with high FOSL1 expression, regardless of existence of therapeutic treatment, had bad prognosis. As regards other breast cancer subtypes than triple-negative breast cancer, statistical significance was observed in some sections, not all, meaning that other breast cancer subtypes can also be treated and targeted genes can be expanded. When talking about totally different solid cancer types, such as pancreatic cancer, renal cancer, glioma, lung cancer, and head and neck cancer, expression of FOSL1 negatively affect prognosis, meaning that the correlation between miR-4516 and FOSL1 may also apply to treatment of other solid cancer types. Let's skip this. All in all, according to our tests using the mimic, the miR-4516 mimic is obviously viable by itself and as a conjunction with the existing drugs, which is why we conceived an idea of the exosome out of either fibroblast or the source, MSC of fibroblast, becoming a pharmaceutical solution. To summarize, we've discovered that proliferation of breast cancer cells can effectively be controlled by exosomal miR-4516 out of normal fibroblast. We've also discovered that overexpressed miR-4516 effectively targets FOSL1 to inhibit growth of triple-negative breast cancer tissues, and that FOSL1 induces bad prognosis in triple-negative breast cancer patients, which is why we think that miR-4516 is a strong, potent candidate of inhibiting at least FOSL1, PAX5, or TCL1A, overexpressing cancer cells. Thanks for listening.